My lecture notes and some slides on the BioCore. I printed some copies, so if you, in case we run out, don't take it if you're not taking the course officially. You're not, okay. All right, so NIH, do you know what that is? National Institute of Health. Actually, they shut down for more than 2 weeks, and they just reopened this morning. So I have a lot of friends working at NIH. NIH is in Maryland, just north of Washington, D.C., about 30 miles north of it. They have a lot of scientists on site doing biomedical research, and most of them had to stay home. NIH also funds a lot of biomedical research outside their campus. So in fact, this university probably receives more than $50 million of research money a year through NIH. And the budget for NIH, annual budget, is about $30 billion. And do you know how much money that translates to per person in the U.S.? Guess or calculate.
$100?
Exactly $100, because we have 300 million people in this country. So
did you find the laser pointer on the desk there? Small? Yes.
Yes.
Thank you very much. Sorry for the interruption.
No problem. So 300 million and $30 billion is $100 per person. So if you are a U.S. citizen, on average, you are paying $100 in your tax money to support biomedical research through NIH. But of course, there are other kinds of research supported through other agencies. So there is a lot of money. Now, the director of NIH appointed by the president, so it's a political appointment. And if you look at the lecture note here, this guy, Elias Zerhouni, I think he was a medical doctor, maybe personal doctor of the father, the senior Bush, and then he was asked to be the director. So he was not a research scientist. So he became the director, and they ran it for several years. And he said that understanding life is the biggest challenge ever faced by mankind. It will define this century, 21st century. And because we are physicists, I suppose you guys are mostly physicists, if you're still in the course. I saw the homeworks, in the lecture notes, pretty physicsy. And so why do we think about NIH? Because I think these questions, the problems in life sciences, are actually too important to be left alone to biologists. Okay? So we need to come and help out. And many of the physics students and scientists can actually really help, because you bring in different types of training and attitude and mindset to the table, and you can make unique contributions. So we often use like 3 different terms to describe the research that physics people do in biology: physical biology, biophysics, and biological physics. And in fact, when I talk to physicists, then I tell them what I do is biological physics to make it sound more like physics. And so my definition is biological physics is yes, it's the study of physics, but using biological systems as a model system. So your questions are physics questions, but the subject of the study is biological. Okay, so you bring the same mindset, same tool sets. Instead of looking at semiconductors, you're looking at cells. So that's biological physics, but you're not really so much interested in the biology itself, but you're interested in discovering new physics in life and things like that. So that'll be biological physics. And so focus is on the, the second word is physics, but using biological materials. So materials physics, it'll be physics, but looking at materials properties, astrophysics, and so on, same here. Physical biology, my definition is that it is a non-trivial application of physics to biological problems. Okay? So it's applied physics, and you're applying physics to solve biological problems. Your discovery will be biological in nature. And I emphasize non-trivial because what's non-trivial today will not be non-trivial 5 years from now. It'll become trivial. So non-trivial means that to be an expert in that area, you actually have to have physical training. Okay, you cannot do it and then do it very well without mathematical and physical training. Okay, that's what I mean by non-trivial applications. But once the technology becomes mature enough, then there can be companies that can commercialize an instrument that you can simply buy and then turn on the key, and then the machine will give the answers for you, and you don't need to know any physics to do so. For example, X-ray diffraction was invented 100 years ago by Braggs, and but 50 years later, people showed that you can use X-ray diffraction to determine the structures of biological molecules such as DNA and proteins. But all the pioneers who actually showed that you can do that were all physicists, right? Because you had to understand Fourier transforms and phase issues and all kinds of things. But nowadays become so routine that if you actually obtain a diffraction pattern from a protein crystal, then the computer will just give you the structure, right, on the spot. Okay. As you take the data in the synchrotron, you can actually see a structure emerging. So then what separates a good scientist in that area now is not physics knowledge. It's actually protein biochemistry. Somebody who knows how to purify proteins in high quality, high purity, in large quantities, and proteins that have interesting properties and so on will win the race there. So there you don't need to know anything about quantum mechanics or light X-ray scattering or any mathematics to do well there. So that becomes trivial application of physics into biological problems, and then that's what I call biophysics. Okay, so again, that'll be non-trivial application of physics into biological problems, physical biology, trivial application, and then this is actually you're studying physical problems for using biological materials. Now, I told you that's what I think, the way things are, but these things are also used very ill-defined way, and so one thing can mean the same thing for some people and so on. Harvard University and Cornell University, they changed the name of the chemistry department many years ago. The departments there are not called chemistry anymore. They are called chemistry and chemical biology. Okay? And, So at some point, maybe some physics department can change the names to physics and physical biology. Although some physicists say that actually that's still physics, so you don't need to add a new name there. All right. So the goal for me for the second half of the lecture is that there are really good seminars going on, biophysics seminars, and physics colloquia with biophysics speakers. And these people tend to give talks at the level that people can understand, targeting at a broad audience. But you still need to know some basics of the terminologies, and also the standard measurement techniques or analysis tools to fully appreciate the presentations. So. And also there are also wonderful papers that are being published in the literature these days on these subjects. And the goal is that by sitting through my lectures, I think there will be 14 lectures, and then doing some homework and so on, doing some reading, you'll be sufficiently knowledgeable to understand the gist of the talks and some of these papers. Okay, so that is the idea. And I never took a biology course in my life. All my degrees in physics, and I teach physics. And in college, I took general chemistry, but never took a biology course. But my work is not entirely biological, and you can do that because biology is easy to learn. I learned by reading a biology textbook that I borrowed from my wife when I was a grad student, and it's much easier than physics textbooks. I cannot read a physics textbook lying on my bed, but I can read a biology textbook. And so it's not difficult, so don't be afraid. And also, because I come from the physics side, I know exactly where most of you are starting from. Basically, you know nothing about biology, so that's what I'm going to assume. And also, if you don't understand anything that I say, that's usually my fault. Okay, it's not your fault. That means I'm not a good lecturer. Just ask me questions, okay, to clarify. And same when you read papers and so on. If you don't understand something, then the writer has not done a good job, right? I want to make you an average reader of a journal paper, or average reader of an audience, average audience member. And once you go through all of this, and then if you still do not understand a talk or paper, that usually means that problem with the authors. Okay, and in biology, when you learn things, my philosophy there was that, okay, I don't understand this yet, but if it's important, it's going to show up again. Then maybe I will study more next time. So you don't have to know everything to move on. Did everyone get my lecture note? Yeah. Okay, so I list 10 papers here, and these are 10 target papers. Some of the papers will be discussed during the class. Some of them will not. But again, the idea is that hopefully by the time you finish, you'll be able to read, most of these papers, and understand the main points without too much trouble. In the online version, electronic version of the lecture note, they can get from BioCore. I also put a link called PubMed link, and you just click, and then it'll go to the US NIH-funded database called PubMed that indexes basically all of the biomedical research papers, for the last several decades. And many of the papers are available actually freely online, and certainly all of them are available if you are connecting from campus, because campus pays a lot of money to publishers to get site licenses. Okay, questions so far? No? No question?
So are you a biophysicist?
Oh, okay. That is a so that is a good question. So I decided just a few months ago, I just called myself a biophysicist, just for simplicity. Okay. So everything is biophysics. Okay. So I want to make biophysics a cool name again. Okay. But that is a good question. Okay. Okay, so let me start with the central dogma, my topic 1. So DNA, everyone knows what DNA is, right? And you know how to spell DNA too. It's DNA. Right? Do you know what it stands for?
Deoxyribonucleic acid?
Exactly. Deoxyribonucleic acids. And so DNA stores your genetic information. It's a blueprint for, for building your body. And so if I make an analogy to a computer, so this will be more like storing information in the hard drive. It's very stable. And the information is still there when you turn off the power, right? But it's actually slow to retrieve the information from, right? And hard drive does not do any calculation, right? What does a calculation? CPU, central processing unit. And in biology, there is a molecule that actually does the calculation. Do you know what that is? Actual actors, players, workers.
Proteins.
Proteins, that's right. So when I was learning biology as a grad student in California, because I had come from Korea, I didn't know any of these expressions in English. So I learned the proteins. I was doing the work, and then when I later, like a couple of years later, I actually learned what that is in Korean, 단백질, and that actually I realized, wow, but that is actually what you eat. This is one of the 3 nutritional elements, right? And so I had no connection between the proteins in the cell and the proteins actually that you eat. So I was actually amazed that, wow, these wonderful machines actually are also what you eat. Okay. So proteins are the cellular players who perform calculations and mechanical work and all kinds of other things. And they are responsible for basically for all of your function, the way you think, the way you dance. All the jobs are done by the proteins. So these are the really important ones. And have you heard of this expression, gene expression? So it's a jargon that biologists use, so you want to be familiar with that. So gene is found in the DNA. It's basically a collection of DNA letters, A, G, C, T. And if you put them in the right order, then it can be the basic information used to make the proteins. Okay? you retrieve information from the hard drive to do the calculation. You retrieve information from the DNA in the form of gene to do functions. The gene expression is like the process of making proteins based on the information contained in the DNA. And this is a very important process because if you look at your brain tissue or your fingernail or your bone, they are made of our own cells. For each cell contains the DNA, our genome. The genome sequence is the same. Basically, every cell has the same DNA. Different tissues, same DNA, but they look so different. Brain is about the softest you can get, right? And bone is the hardest tissue you can get. They are so different in physical properties and size and their functions, despite the fact that you have the same information stored in the hard drive. Okay? So why are they so different? Because different proteins are expressed at different levels, at different times between different tissues. Okay? So where and when a gene is expressed, it really determines what happens to the cell and what happens to your body. Okay? So that is called regulation of gene expression. So you want to make proteins based on the genome, but you need to know when and where certain proteins are produced. So you have to regulate when that happens, where that happens. So that's gene expression regulation. That's a very important process. So we are not that much different from chimpanzee in terms of actual proteins that we produce, almost identical. But we can be very different from the chimpanzee in terms of where and when these proteins are produced. So we differ much more from chimpanzee in terms of regulation of gene expression than the genes themselves. Okay? Now, you cannot make proteins from DNA directly. You have to go through an intermediate, just like when you retrieve information from the hard drive. To send to CPU, you have to have RAM, random access memory. Because now, with smartphones and so on, all these things are kind of getting blurry. You don't have a hard drive on your smartphone. You have what do you have? You have SSD, FRAM, or things like that. Anyway, but this is a temporary storage of information because if you turn up the power, it disappears, right? It's erased. So likewise, you have an intermediate called RNA. And RNA stands for ribonucleic acids. So deoxyribonucleic acid is actually RNA, but after you remove an oxygen atom. Okay? So that's the difference between DNA and RNA. The process of making RNA from DNA, that process is called transcription. And there is a cellular machine called the RNA polymerase. That is doing the transcription. Okay? So RNA polymerase can come and use a DNA as a template to make RNA. Then the process of making proteins based on RNA is called translation. And ribosome is the cellular machinery that is making the proteins based on the RNA sequence. And you need to have all of them to sustain the life, as you know it. And this is called the central dogma of molecular biology. Okay, you have DNA, and you make RNA, and you make proteins that perform functions. That's a central dogma. Of course, dogma is there and to be broken, right? So there are some exceptions that we can discuss. It should be emphasized that RNA polymerase is also a protein. Okay? And ribosome has a lot of proteins in there too. So to make actually proteins, you must have proteins too, right? So that when the life first began on this planet, it could have begun on Mars, right? But it started somewhere. How do you start when you need to have all of them at the same time, right? It's a chicken or egg question. What came first? Right? If you need to have proteins to make RNA, in fact, you actually need to have proteins to make DNA too. So you make DNA based on DNA sequence. So that is called replication. It's done by a protein called DNA polymerase. But that's also a protein. So to make copies of DNA before the cell divides, you also need to have proteins. So then really, how did it all happen, right? So that is a very interesting question. It's the origin of life question. Okay, questions so far? Yes.
Pardon if this is not a good question, but is there any charge difference between DNA and RNA?
Charge difference? No. So the DNA and RNA molecules are what's called polyelectrolytes. They carry negative charges. And one unit of DNA or RNA has one negative unit charge.
Okay.
Yeah. And their electrostatic properties are very important. Turns out that if you have two unit charges separated by that separation inside the DNA or RNA, and if you put that pair of charges into water, with a dielectric constant of 80, and you calculate the energy of that electricity interaction, that energy matches the thermal energy. Okay? I'll come back to that later, but what that means is that whenever some energy scale in biology matches the thermal energy scale, then it's important for biology because this actually can change and on the relevant time scale for life processes. Okay, on this part, there are actually several Nobel Prizes. Do you know any of them? Huh? Watson and Crick, structure of DNA. So Watson and Crick, they were student and postdocs in Cambridge, UK, in the 1951 tour and so on. Actually, Watson was an American who got his PhD from Indiana University as a biologist, and then he went there as a postdoc, and he was young. He was like 25. And then Crick was actually a PhD student who is a physicist, but he had served in the UK Navy during the Second World War, so he was much older. He was like 35. So you had like 10 student who was 10 years older than the postdoc, but they shared the office and they decided, okay, let's solve the structure of DNA, and they shared the Nobel Prize 10 years later. So that is the first DNA Nobel Prize. There's actually another prize here too, on the DNA side. Arthur Kornberg, I think he died just a couple of years ago. He discovered DNA polymerase. Okay, and he isolated protein sample from a cell that has a DNA synthesis activity, and he won the prize for that. Anything else?
Transcription.
Okay, so I know one prize for transcription. In fact, I don't think I know anything else. There was a transcription Nobel Prize about 6-7 years ago by Roger Kornberg, who is the son of Arthur Kornberg. So the father won the prize for discovering the DNA polymerase, and son won the Nobel Prize for determining the structure, atomic structure of the RNA polymerase. Okay, and he's still alive. Translation, I know 2 prizes at least. A few years ago, there was a prize given to 3 people for solving the 3-dimensional atomic structures of the ribosome. And their work was done in 1999 and 2000, and 3 of them shared the Nobel Prize in chemistry. So actually, I think the chemists are actually getting a bad deal these days, because the Nobel Prizes in chemistry now, at least half of them are given to biological work, right? So structure of the ribosome, why is that chemistry? But the Nobel Committee claims that by looking at the detailed structure of the protein-making machine, you can understand the chemistry of the peptide bond formation, okay? And same for RNA polymerase structure, it was a chemistry prize. I'm sure Klaus Schulten mentioned some Nobel Prizes. Many of them are given in chemistry prizes. This is because chemists are actually stupid, right? Physicists are really smart. So none of the Nobel Prizes in physics has been given to biological work. Although there are many biophysicists, there are many physicists who are doing biological work, and some of them could be called real physics, but 0. Because they know in physics community, once you open the door, then you cannot stop the flood. And the chemistry community, they have made a mistake. So, if you do not allow these kind of biological prizes, then a lot more real chemists can win the prize, but it's too late. And there's another prize for translation. It's called it's a prize on genetic code. So the idea is that, if you know the sequence of the RNA, then how do you translate it into the component of the protein, which is amino acids? Okay, how do you make, how do you actually encode the information in terms of, nucleic acid letters into, proteins? So that is a genetic code, and there was a prize for that too. In fact, one of them who won the prize was working in NIH. Do you know a physicist named Gamov? How do you spell it? I think he's a Russian physicist, right? Gamov. In fact, when I was in high school, I read a story written by him. It was some Mr. Thompson, going on a space trip. So he actually popularized physics too by writing short stories. And so there's actually a book written by James Watson, and the title is, I forgot the order. I mean, I read it many years ago. So the title is basically 3 words, but in some I forgot which order. Okay, so basically this is James Watson wrote a book called The Double Helix to describe, the surrounding stories around, DNA structure discovery, and then many years later, he wrote a book. I think this is maybe about 10 years ago, where he then tried to, search for the genetic code. stored in the RNA. So that's where this comes from. And then when he made that Nobel Prize-winning discovery, Francis Crick's wife told James Watson, well, now you are now famous. Now you should get married. So that he was chasing after girls, and then he also got to know Gamow, because Gamow studied RNA necktie club. Do you know? Have you heard of this? So there were 20 scientists that Gamow collected, and they were all interested in the genetic code. So what they did was they made RNA necktie, each one, but because there are 20 different amino acids, right? So they made one type per amino acid, and then each one of them took one. Anyway, so that's why the name this comes there. But Gamow didn't win the Nobel Prize. All right. Now let me tell you about another Nobel Prize that actually potentially solves this, chicken and egg problem. So have you heard this expression, RNA world hypothesis? So Tom Cech and Sidney Hartman, they won the Nobel Prize, and for discovering what's called the ribozyme. Before I explain this, I need to tell you what an enzyme is. Enzyme is a protein that catalyzes biological reactions to make certain reactions much faster. And the first enzyme that was named is urease. And the rule is that you add ASC at the end of a word to show that it is an enzyme that destroys whatever is in front of it. So urease was an enzyme that destroys urea. Do you know where you find urea in abundance? Yes. Anyway, so that's the naming scheme, and DNA polymerase is an enzyme because it ends with ASC. Okay. But it's kind of strange because what the DNA polymerase does is, if you have a polymer of length n, that it makes it longer, right? That's what the polymerization reaction is. It makes into a polymer that is longer. So then how do you call it an enzyme that destroys things, right? That's strange. So for physics students here is that you have an operator that destroys and then creates another one, right? So in a sense, you are destroying a polymer of length n to create a polymer of length n plus 1. So I think you can call this polymerase too. Okay. And other enzymes are like ATPase. So ATP is a chemical energy source that is used to power the cell, most of the life processes. And ATPase is an enzyme that destroys ATP, in this case to make ADP and the phosphate to release the chemical energy stored. So these are the enzymes that we'll get to discuss. So ribozyme break the rule because I said enzyme is a protein that catalyzes biological reactions, but ribozyme is actually is an RNA enzyme. It is an enzyme, but not made up of proteins, but made up on RNA. Okay. So this discovery was really surprising, because until then everyone thought that only the proteins can function as enzymes. Okay. Now, then this really raised an interesting possibility. DNA is the information storage. Protein is the cellular plate that perform functions like enzymes. But you know that RNA can also store information. In fact, some of the viruses, their genome is encoded in RNA. HIV for AIDS, its genome is in RNA. Your influenza virus that infects you seasonally, its genome is also in RNA. So RNA clearly has the ability to store genetic information. So if the RNA can perform functions, right, and store genetic information as in RNA viruses, then you don't need DNA, you don't need proteins. You only need RNA at the beginning. Okay? So the idea here is that at the beginning there was RNA, okay? And the RNA was sufficient. RNA could reproduce because it can function as its own template and also its own enzyme, as its own polymerase. Later on, to make the storage more robust, just like having a hard drive, somehow evolution invented DNA as a more robust storage form. In fact, that's true. The RNA is actually much more labile than DNA. Okay, when people sequence the genome of the DNA, Neanderthals from Europe, right? They are looking at DNA, remaining DNA, but RNA is long gone. Okay? RNA is much more stable. And later on, proteins are also invented because proteins perform much more diverse functions, and also they can be much more efficient than RNA enzymes that are typically limited in what they can do and the efficiency of their operation. So that's the idea. You start with RNA, and then DNA and protein were added later on. So that's another part. That's what most people now these days believe. Questions? Things pretty clear? And there is another exception to the central dogma, and also resulted in another Nobel Prize. So the flow of information is from DNA to RNA to proteins, and DNA upon itself. And basically, according to this, you cannot make RNA based on protein. You cannot make DNA based on RNA. Okay, that's the dogma. But this is still true. There's no known example where can make RNA based on protein out there. Okay? But it is possible to do so. Okay? And that process is called reverse transcription, because it reverses the process of transcription. Okay? Now you're making DNA based on RNA, and so enzyme that does job is called reverse transcriptase. And you can see that they're not really following my rule. It's not destroying transcription per se, right? So oftentimes people get lazy. They just add A's to whatever function they want to study. Like if an enzyme is elongating a filament, then that's called elongase, or things like that, right? And there was a prize, big Nobel Prize for this too. HIV encodes a reverse transcriptase. In fact, what it does is that it infects your cell, and then it makes a DNA based on the RNA genome, and then it then inserts the DNA into your own chromosomal DNA, so that the virus can lay dormant for many, many years before it comes out again. Okay? And that's called a retrovirus because you are doing the process of making DNA based on the RNA. In our own body, humans, we do not have a reverse transcriptase. It's only, as far as I know, it's only found in viruses. But this became a very important tool for biotechnology, because if you want to, analyze what kind of RNA molecules are produced in the cell or tissues, you need to quantify the number of RNA molecules. Typically what people do is that they do reverse transcription of your sample to make DNA and then analyze DNA, because DNA is much, much easier to analyze using DNA sequencing and so on. Yeah, that's another Nobel Prize. Sanger and others got the Nobel Prize for discovering a method to sequence the DNA. Do you know Moore's law? What is that?
Isn't that the one where the size of a particular element of circuitry is going to decrease every 10 years? It's cut in half every 10 years or something?
Actually, I don't know. I don't know the precise value. So it's like size of the feature in a CPU computer device is transistor, or not density of the transistor in a chip, or the speed or cost, and everything is actually changing exponentially, and like doubling every 3 years or 5 years. I don't know, but that has held up for a long time. Now, the cost of DNA sequencing actually is Weber or ultra or super Moore's law, because actually it's not exponential. It's actually more than exponential. And so original sequencing method is no longer used, and the new methods can sequence your entire genome for a couple of $1,000s these days. Although I think they spent $2 billion to sequence the genome of James Watson. Okay? So that's really striking. And maybe I'll have a chance to talk about sequencing technologies. I know that about 15 years ago, 2 assistant professors of chemistry in Cambridge, UK, they decided to, let's start a company to sequence DNA molecules at the single molecule level. And they raised a couple $1 million, like a couple £1 million. And when they talked to me about their company back then, I thought, oh, you guys are crazy. It's never going to work, right? And then the company actually changed the direction, to go away from single molecules to some amplification-based method. And then eventually the company was sold to a U.S. company for $600 million. And the company is named Illumina. It's a dominant sequencing company. And their market value is now $10 billion. And that's entirely based on that technology invented by 2 junior professors in Cambridge. So it's quite really remarkable. And people now use DNA sequencing to perform diagnosis of Down syndrome. From fetus, it's actually from mother's blood. Because mother's blood contains some DNA from the fetus, by just doing the sequencing of the mother's and fetus DNA, a lot at low cost, we can actually tell with 99.9% certainty whether you have extra copy of the chromosome in the baby or not. And so now, people do not do this invasive process anymore, and you just pay, some money and then sequence mother's blood, and then all these technologies actually are really benefiting in the end us and hopefully improve the human conditions. Okay, so let's take a break, 5 minutes, and we'll come back and continue. But any question? All right. You took this course a long time ago, as an undergrad, right?
Yeah, I didn't know it was. Is it the same? The paper.
Papers have changed, yeah.
Yeah. Oh, I looked it up, and the spelling of gamma seems to be W at the end.
W, okay. Did you look up the book?
Oh yeah. Yeah, it was showing up.
So in the book, he talks about one woman he was really chasing after majorly, and she was a daughter of a really famous evolutionary biologist in Harvard University. I forgot actually, the name doesn't come up to me, but.
The order seems to be genes, girls, and gamma.
Okay, genes, girls, and gamma. Okay. So when the humans first came out of Africa, maybe 100,000 years ago, the gene expression of an enzyme called lactase, I hope that's the correct name, was pretty straightforward. Okay. Lactase is an enzyme that digests sugar called lactose. Lactose is a sugar found in mother's milk. Okay, so that's what makes milk taste very sweet. That's lactose. And so for a baby to digest milk and get nutrition out of it, you need to have a lactase in the baby's stomach and so on, right? But then once the human child becomes like 7, 8 years old, then you no longer get milk from the mother. So it actually caused extra energy to produce lactase enzymes in the body. So it doesn't make sense to continue to produce lactase, right? So then the evolution dictated so that once you are 7, 8 years old, then you don't express lactase in the body. That makes sense, right? So you don't want to make things that are not needed. That changed actually when the agriculture started in the Middle East. So there's a mutation arose in lactase, surrounding area in the DNA that continued to produce lactase, because that had a selective advantage, because people began to, herd cows for milk. And then if you actually have a mutation so that you make lactase as an adult, then you can utilize another source of nutrition, and then you can actually survive better, right? So that's the idea. So if you're Caucasian, then you have the mutation. I think 90%. So, but if you are from Eastern Asia, then it's just the opposite. Only 10% people have the mutation to produce lactase as an adult. So what that means is that if you drink a pint of milk, and without expressing lactase, then you get severe diarrhea and also a lot of bad things. So actually, my grandmother-in-law, whenever she had constipation, apparently she drank a glass of milk, and then it solved the problem. So actually, I don't know if I have the mutation or not. So one way to test is next class I can bring a glass of milk and then drink at the beginning, then see what happens. All right. But if you can drink a glass of milk and then survive fine, without any side effect, then you are still producing lactase. Okay. And somewhere in Europe and Eastern, Middle East, there was a strong selective pressure to favor this mutation. Now, the reason I'm talking about this is the following. So the mutation is not on the gene itself. So if this is a DNA, and this part of the DNA is a gene for lactase. When RNA polymerase binds and then makes RNA, and then RNA is converted into an enzyme called lactase protein. That's what is used for copying. But the difference between this lactose tolerant mutant and so on is not here, but just outside here. Okay, that's called so by changing the sequence outside the gene in the regulatory region, then you can change the gene expression pattern of that gene, where and when the gene is expressed. Okay? Of course, you don't make this lactase in your brain, because I guess you don't get sugar there. You have to express in your stomach. But when do you express? Well, until you are 7, right? That you don't need to drink milk anymore. But then, when there's mutation arising here, then you keep making this. You don't turn it off at the age of 7. You keep making it. Okay? So that's the idea. So when evolution selects the useful traits and so on, usually selecting based on the regulatory changes is much quicker. You don't need to make a new protein, but you simply change when and where these proteins are made or expressed. All right, so again, I told you that the regulation of gene expression is very important. It tells you why we are different from chimpanzee, right? And in fact, I have a quote here in the middle of that second page by somebody who I can name because I don't know who said this. But if you give me coding sequences of a rat, I mean, if you give me the gene of a rat, and then the regulatory sequences of a monkey, I will give you a monkey. Okay? If you make a chimera DNA between, gene of a rat and the regulatory sequence of a monkey, then what you will get is a monkey, because this is what really distinguishes between different animals, not the gene itself. And of course, it is a very strong statement. There are exceptions, but to the first order, this is a correct statement. Okay. So it's important to understand how the gene expression is regulated. And there was another Nobel Prize for two scientists who actually did the first study of gene regulation in molecular biology. And what they did was to study how the lactose is used by a bacterium, not by human, by a bacterium. And this bacterium is called E. coli. And you know that E. coli is, sometimes bad. You can have an E. coli outbreak that is pathogenic, from the spinach farm or tomato plants, whatever. It normally lives in your stomach, right? So it's gut bacterium. And E. coli is, kind of elongated. So it looks like a chalk, or if you want, puffy Cheetos. And it's spherical here, and the diameter is about 1 micron or slightly less, and then this can be 3 microns in length. E. coli loves sugar, and it actually loves glucose the most. So if you give the E. coli glucose, then it survives on glucose. Okay, that's the energy source. And it doesn't like lactose. And I'm not a chemist, so I don't know how to draw the structure of the lactose. But glucose is a simple sugar, 1 ring, but the lactose has 2 rings. Okay, so it's a bigger one. So to utilize, you have to go through extra steps. So so E. coli does, if you give E. coli both glucose and lactose, then it's not going to use lactose. It's going to use glucose only. Okay, but when glucose is absent, and then when you have lactose, then E. coli, what it does, it actually makes an enzyme that can digest the lactose. Okay, so it's actually somewhat related to what I described in terms of I guess my unpleasant story. So E. coli has an enzyme called and people just call it beta-gal, so I'm going to do that too. So beta-gal is an enzyme that can break lactose into smaller sugar. Okay? But when E. coli is happy with glucose in the surroundings, then E. coli doesn't need to make beta-gal, right? So, and if there's no lactose around, there's no need to make beta-gal. So in fact, E. coli does not make, this enzyme, almost negligible. But when suddenly there's a lot of lactose coming in, then it'll have to make the enzyme to survive. So the way it works is the following. So you have that's DNA of E. coli, part of it. And then this is the gene for the beta-gal enzyme that is required to break lactose. there's a small piece of DNA sequence called lock-up operator. What happens here is that there is a protein called lock repressor. And this protein, I'm going to draw it, this manner. So this lock repressor binds to this special sequence under normal conditions. So it binds there. And because it's blocking the binding site for the RNA polymerase that needs to copy, this sequence into an RNA, so transcription cannot happen because lock repressor is bound there. So that's why this is called repressor, because it represses the expression of the RNA. So when lock repressor binds to that sequence, then this gene cannot be expressed under the normal conditions. Now, when lactose level actually increases, this lactose as a small molecule can come and bind to the protein lock repressor, and then it's a small protein, a small molecule that binds to a large protein, but maybe is tickling, and then the protein undergoes a shape change, and the jargon there is a conformational change. That's what biologists use. When they don't know what's going on, they just call it conformational change. And so basically, small molecule binds here, and then it induces a shape change of the protein, and then protein no longer binds to the DNA tightly, and then dissociates. Okay? So when the protein is gone, right? So you have this lactose-bound protein is leaving the DNA, and then now to the freed up space, RNA polymerase can come and bind, and then start transcription. Okay, so that's really beautiful, right? Only when you have lots of lactose in the surroundings, then this repressor will dissociate to allow the expression of the enzyme required to digest lactose. So this is really like in a nutshell how gene regulation works in the simple model system. Yes.
So the DNA must be in the nucleus of the bacterium.
Mm-hmm.
That means the lactose must go into the nucleus to make
Yes.
this process.
Yes. Yeah. So he raised a very important point. But before I answer this, do you have any more question about this? Does that make sense? That there's a feedback system, so when you have nutrition, then that nutritional molecule itself is actually binding to the repressor to cause it to dissociate, and then RNA polymerase can come and make the RNA, and then later into proteins. So that's really nice. Basically, you don't have to have an extra person sitting there telling them what to do. They can, it's all, self-controlled, right? That's great. I told you that this is beta GAL, but there's also another gene here, and this encodes so this is gene lactose permease. So what does permease mean? It's an enzyme that permeates lactose through the cell membrane. So again, this is they didn't follow my rule, okay? But I didn't make this okay. So when the bacterium decides, I don't have enough glucose and there's some lactose around, then I want to take up as much lactose as possible, right? So then this also results in the production of lactose permease that goes and binds to the membrane, and then to allow the influx of lactose into the bacterium. Okay? And there's yet another enzyme there, but that's the details. So it's really a beautiful system that people really have figured out over the years. And this is a really prototypical model system, if you want to understand gene expression this is something that you first read about. Yes?
So if you want the permease to be produced, we must first make the lab repressor
Mm-hmm.
to be away from the DNA, and
Mhm.
that requires lactose.
Mm-hmm.
So it's
Second chicken egg problem here, right? So, so there, the lactose, does not, it's not like it doesn't come in at all, right? But this makes the permeation much faster, right? So if you have really high level of lactose outside, then you will still feel it, right? And then looks like I have lactose out there, and then okay, let's take them in, right? So E. coli is a really great model system. Someone said that, actually in biology people use several different model systems to study. Because you want to study human, but human, it's not so easy to do genetic manipulation and so on. If you want to understand, what certain gene does, and you want to make an animal model where you remove that gene and then see what happens. And so people then use a mouse as a model system because a mouse has a short lifespan. You can do genetic manipulation very easily. And then if you want to do it even simpler, you can go to a zebrafish. It is a model system for vertebrate animal, although it's not a mammal. So there are many model systems, going down to fruit flies and nematode and yeast and so on. So different labs use different model systems. But people say that, well, if you want to, understand, that there are two cells you want to understand. Two. One is your model system, and then E. coli. Okay, so it's really such a standard system that basically, one should really have a pretty good knowledge of E. coli, even if what you're studying is plant biology, because there are a lot of parallels between different systems. All right, so I want to move on to the second topic. It's really good to know how big different things are, right? If you know that the E. coli is 1 micron in diameter, then you would not say that, well, ribosome could be 10 microns in size, right? Because ribosome must be inside the bug, right? In fact, single bacterium can contain several thousand ribosomes, and ribosome is about 25 nanometers in diameter for the ribosome. A typical protein, has a diameter of about 4 nanometers. And some proteins can be much bigger, but like an average protein would be just a few nanometers in size. DNA, in the double-stranded DNA form, the diameter is about 2 nanometers. And so on. So many of the biological molecules are in the nanometers in size. So really, at the molecular level, just few nanometers. Okay, so that's at the molecular level, at the level of single proteins and RNA, DNA, and so on. The important physical length scale is just a few nanometers. The relevant energy scale in biology is Kb times T, the thermal energy. So Boltzmann constant Kb is 1.38 times 10 to the minus 23rd joule per Kelvin, and temperature at room temperature is around 300 Kelvin. So and at the ambient temperature, then this is about and we know that joule is equal to Newton times meter. Okay, so that I can rewrite it as 4.2 times So I divide 10 to the minus 21 to 10 to the minus 12 and 10 to the minus 9. Okay, and then put Newton there, and then meter there. So this is a piconewton. You know how it goes down, right? You have meter, millimeter, micrometer, nanometer, picometer, femtometer, attometer, zeptometer, yoctometer. Oh, you don't know this? Okay. You don't need to know. You only need to know down to about femto, but femto is 10 to the minus 15. This is 10 to the minus 12. And this is 1 nanometer. So the important energy scale in biology is kT. That is about 4 piconewton times nanometer. Okay. Why is it important? Because it goes into the Boltzmann factor, right? When you have thermodynamics or rate equations and so on, in the Boltzmann factor you have this. So basically, when the energy scale is about this, you get the biggest effect. And in biology, energy scales that are much, much larger than this, not important because they don't change, right? Through thermal fluctuations. Things that are much less stable, they change too quickly, they're not important. So things that are about kT are what's important in biology, what matters in biology. Now, I told you that nanometer is important length scale for molecules in biology, and kT is the important energy scale in biology. Then what will be the important force, mechanical force scale for molecules? Piconewtons. That's right. So piconewtons is do you know how big a piconewton is? Do you know how much my phone weighs? in terms of Newton?
Well, about 20 piconewtons is enough to denature a protein, right?
Wow, you are using a jargon, denature. Okay.
Um,
About two. I think it's more. Usually, maybe 200 piconewton or higher. So Newton discovered his gravity, based on apple falling from the trees, right? So if you have a very small apple, about 100 grams, the gravitational force on that apple is a Newton, right? Because what is it? 9.8 times, whatever, 0.1 kilogram. That's 1 Newton. So this is about actually a 1 Newton. So piconewton is, of course, that much smaller than that. Okay, so this is a good time to stop, and I'll see you on Tuesday.